This is part 3 of the 100,000 Genomes project lecture. So you've seen the sequencing technology. We're now looking at how this data has been used. The project was based upon trying to detect both genetic abnormalities in cancer, and we're going to talk about the genetics of cancer next week. So I'll give you more detail on the sorts of things we're looking for in that lecture. I'm going to focus more upon the clinical genetics side of things and what sort of things the 100,000 genomes are looking for in what we think are likely to be patients who have got a hereditary disease or a disease with a genetic basis. So we looked at patients who present with rare, unusual genetic disease or genetic syndromes or just unexplained disease. And this is a linking to one of the early success stories where patients have been identified, so have a little look at that. Now what this sometimes shows is that an affected child has got a disease and the same mutation is present in the parent. And that's fairly straightforward. You can see that it's a classically inherited either autosomal dominant, autosomal recessive or X-linked or some other inheritance pattern. But sometimes both parents are negative for the mutation, but the affected individual is positive for the mutation. And what we find in those situations is these are what we call de novo mutations, and these are mutations acquired in the germline. So this has come from a mutation that's cropped up in a sperm or an egg, but is not constitutionally present in the parents or in the siblings. And if you find a de novo mutation, you know that the siblings are probably very unlikely to be at risk of this same disease. Now what we're finding is de novo mutations are particularly relevant for large genes. Just the fact that large genes are more likely to pick up a mutation by random chance than a small gene, and there are lots of very large genes controlling brain development. And that partly explains why we find de novo mutations in a subset of autism and autistic spectrum disorder, schizophrenia and other neurodevelopmental diseases. And this link here is worth having a little look at to see the frequency of these. So this is this study which I've just opened up, and it's looking at the frequency of de novo missense mutations and just other de novo likely gene disrupting mutations, so things that will cause premature stop codon or change one amino acid into something very different, or maybe alter a splice site or frameshift mutation, that type of thing. So this particular group did whole exome sequencing. Now whole exome sequencing is just basically taking the RNA and reverse transcribing that into DNA and then sequencing that. So whole genome sequencing is starting with DNA, whole exome sequencing is starting with RNA. But the end result of this is looking at autistic spectrum disorder. We're looking at up to 43% of cases having likely de novo mutations that were not present in the parents but had acquired due to a genetic mutation in either the egg or the sperm. What is really interesting and what's coming out of this type of study, and this is quite old now, this is 2014, lots of studies have been done since, but there tends to be an overlap between the genes that are affected in males that seem to cause autistic spectrum disorder and in females that may cause other disorders including schizophrenia. So the same genes seem to be involved but having a different clinical effect, depending on whether those mutations occur in a male or in a female. So this is summarised here. We've got some overlap between autism and other neurodevelopmental diseases in that the same genes seem to be affected. However, it's very difficult to say for certain whether these mutations that have been picked up are absolute proof that they are disease causing, because you've only got one individual with that mutation who happens to have the disease, and it may well just be a random mutation that has cropped up. So it is quite difficult to be absolutely sure these are disease causing. They're just called likely disease causing. What we are finding with autism and autistic spectrum disorder is a whole range of clinical symptoms that are observed, and what we find is mutations in these likely disease causing genes. If you have identical twins and siblings with the same mutation, you tend to have individuals with a similar phenotype. However, if you find two siblings who both have autistic spectrum disorder having different de novo mutations, and this does happen, it happens rarely, you find that these have very differing symptoms, adding weight to the fact that these mutations are likely disease causing. So this is summarised here. We're looking with typically large genes. Large genes are more susceptible to de novo mutations than small genes, just by random chance. These likely disease-causing genes are CNS expressed. What we often find is that both of the parents are normal in terms of normal for the sequence of that particular gene, and the siblings are, when I say normal I mean wild type, siblings are also wild type for that particular gene. So the individuals show a unique mutation not present in the parents or the sibling. Now this high frequency de novo mutations might also explain why the genes that are affected, or the genes that are cropping up in this sort of study, are not genes highlighted in genome wide association studies. And obviously that should have said genome wide association studies, and I've just corrected that typo. So genome-wide association studies look for linkage between parents and, well, they track inheritance of specific regions of DNA between parents and offspring and see whether that tracks with the presence or absence of disease. So genome-wide association studies are very good for finding hereditary mutations that cause disease. However, a disease that's caused by de novo mutations would not be picked up by genome-wide association studies because there is no intergenerational linking of the mutation from one generation to the next. Here's just another study, again looking at de novo mutations in individuals with schizophrenia. In this case they found 4 out of 15 de novo mutations which were nonsense mutations, so we're expecting those to be gene disrupting mutations, and this is much higher than what you'd expect by random chance. So this group are suspecting that those particular mutations are disease causing mutations. And these are all being picked up by whole genome sequencing. Okay, I'm going to move on to some bioinformatic issues and then come back to some ethical issues towards the end. So bioinformatic issues relate to the quality of the DNA sequence and whether that is useful, and sometimes the sequence comes off the sequencer and doesn't directly match to anything in the human genome, and in many cases you get random bits of sequence and you just disregard these as junk DNA, and this is based upon some computing algorithms that are written by bioinformaticians, and they decide what is good, what is not good, how much of it has to match the human genome, how much of it doesn't. If it doesn't match in a certain way, it just gets put down as junk DNA. However, what we find in some disease situations, particularly in cancer, but also in other situations, we get things called indels, which are short insertions or deletions. These can be caused as an artifact of sequencing. Equally, they could be disease-causing mutations. So it's difficult to know whether these are genuine sequences or whether they are artifacts. And this is where bioinformaticians write lots of coding programs to try and figure out what is the best thing to do with these sequences that don't directly match the human genome. I suppose the whole point of doing sequences is we're looking for stuff that doesn't match the human genome in small bits, but you don't want to then assign lots of junk sequencing and claim that they are mutations. So this is one of the big bioinformatic problems that bioinformaticians have to figure out. Now, one example where a DNA sequence was almost certainly put into the junk DNA when it shouldn't have been was in the identification of a very abundant translocation in prostate cancer. So I'm going to talk more about genetics of cancer next time, but there was a translocation that occurs in up to half of all prostate cancers between the TMPRSS2 gene, which is an androgen-driven gene, it's a serine protease, and the ERG oncogene. And the ERG oncogene drives cancer growth. And what's effectively happening is we're getting an intrachromosomal deletion, which basically loses the end of the TMPRSS2 gene, so it deletes that, and it deletes the start of the ERG gene, and we get a fusion of exon 1 of the TMPRSS2 gene with exon 4 of the ERG gene. And whole DNA sequencing didn't seem to pick this up, maybe because it just saw a sequence and then it suddenly changed into something else and it was classed as junk DNA. But when we did when it was analysed by transcriptomics, that is taking RNA, sequencing that, what was found is this was quite an abundant transcript, and it was repetitively seen again and again in multiple cancers. So the whole genome sequence in a prostate cancer didn't initially flag this up as a translocation. It's likely a lot of the sequence, because of the algorithms that were written, the DNA was sent into the junk folder, but when whole exome sequencing was done, this translocation was fairly rapidly picked up, because we've got overlapping reads here from the ERG gene, and then we've got TMPRSS2, and this is exactly the same mRNA sequence in almost every prostate cancer that has this translocation, whereas at the DNA level, the translocation occurred in a different place somewhere between exon 1 of TMPRSS2 and exon 2, or just anywhere between exon 1 of TMPRSS2 and ERG exon 4. Now, this region here that is deleted is about 3 million base pairs. So if this stops somewhere after, or somewhere before exon 4, and this stops somewhere after exon 1, you wouldn't naturally pick up that this is the same intrachromosomal deletion happening in all of these prostate cancers. It's only when you look at the transcriptomics you see that lots and lots of different deletions in roughly the same place give rise to exactly the same mRNA sequence. So here, whole exome sequencing picked up this translocation much more efficiently than whole genome sequencing. Now, another thing that another bioinformatic issue is repetitive sequences. So wherever DNA is repetitive, it's very difficult to know exactly where it fits on the human genome, because the same sequence exists lots of times. So I showed in video 1 that we could blast this sequence here and it appears only once in the human genome, whereas if you blast this sequence, it will occur hundreds of thousands of times. So you've got repetitive sequences which are present more than once in the human genome. And in addition, we've got pseudogenes and retrogenes, which have very strong sequence homology with normal, proper genes, I'll call them. So this can cause problems of, retrogenes are often base for base identical to the true proper gene from which they were copied. So this causes lots of problems. Other problems come from things like the HLA region, also known as the MHC region, which is a highly repetitive sequence that determines our tissue types. And there's lots of genes that are very, very similar to each other and very repetitive. So whole genome sequencing is really quite rubbish for getting an accurate sequence across repetitive sequences. And this is nicely summarised here in terms of the difficult to read regions. The HLA region, highly repetitive, highly polymorphic. It's used for tissue typing and transplant matching. And when this is done clinically, it's done using a targeted sequencing approach rather than whole genome sequencing, so targeting specific regions and genotyping specific regions. And this is because the confidence of the sequence, you need a read length of 100 and you still don't get up to 100% certainty that that is in the right place. So particularly with short reads, you're really not sure exactly where this particular piece of DNA comes from, whereas even with quite long reads you still can't be sure, because you might have stretches of DNA that are over 100 base pairs in length and you cannot have any certainty as to where on the HLA region that came from, because it is so repetitive. So you have to go down other approaches, targeted polymorphism and mutation detection, either by genotyping methods or directed PCR methods or directed sequencing methods. And this is summarised down here. This is a study from the 1000 genomes study from quite a few years ago, and it really highlights that 1,000 genomes, whole genome sequencing data sets are basically unreliable and unsuitable for determining a patient's HLA type. You need to do targeted sequencing or targeted genomic mutation detection using specific primers, and in general the whole HLA region is omitted from lots of whole genome sequencing projects. Now, if you want a sequencing technology that can read across the whole HLA region, it's worth having a look at nanopore sequencing. Nanopore sequencing can do very, very long reads of maybe 100,000 base pairs start to finish. I'm not going to cover it here, but for those who are interested in DNA sequencing, just have a look up at nanopore sequencing and see how it could potentially solve this problem. Okay, so I'm going to get on to some ethical considerations. There are a few more slides on the PowerPoint that you can just have a little look through. I think they were mostly self-explanatory, but I'm going to focus on some of the ethical considerations now. For people who took part in the 100,000 genome studies and also all of the other big sequencing studies, there's been a recent announcement that the government might be trying to sequence a few million genomes now. If you take part in such a study, someone may find out any of the following. And when I say someone, initially the bioinformatician, who will then code your genome and flag up all of the risk alleles. It will get the whole genome sequence, match it against a reference genome, and then flag up all the bits that differ or are, shall we say, interesting. So you could carry an ApoE gene allele called E4 allele that gives you a 20-fold increased risk of Alzheimer's disease. Would you want to know that, or would you not want to know that? I mean, if you do want to know that, you can take a consumer genomics test, Ancestry.com, and that genotype is on there. So it's the sort of thing that you could find out if you really wanted to, and it would also be on some sort of report from a whole genome sequencing point of view. This is, just a risk. It's not a certainty you're going to get Alzheimer's. You could be told that you're a BRCA1, BRCA2 carrier, which means you're likely to get breast or ovarian cancer by the age of 50, and maybe 1 in 1,500, 1 in 2,000 people may have these mutations and not know about it. So you could enter into a whole genome sequencing study for something relatively innocuous or something completely unrelated to cancer, and then someone may find out that you are genetically predisposed to cancer. The question is, would you want to know that information? It could turn out from your whole genome sequence that you carry a rare, lethal, recessive genetic disease. And that sounds quite alarming. Don't be alarmed, because most of you carry 2 or 3 of these. So cystic fibrosis is one of them. If you're from Northern European Caucasian descent, about 1 in 25 to 1 in 20 of you will be carrying cystic fibrosis. Maybe 1 in 30 might be carrying something like MCAD. So there are lots of these out there. Most people carry a few lethal recessive mutations, so it's probably not worth telling you about this. Something that could come up on this is you could turn out to be homozygous for much of your genome. It could mean that you are the offspring of a very close consanguineous relationship, maybe offspring of cousins, perfectly legal. However, in that situation, if you carry a rare lethal recessive genetic disease, the chance of your cousin carrying that could be 1 in 8, and therefore, the chances of a child having this disease could be really quite high. But it could also flag up that you could be the offspring of an incestuous relationship. And if you are homozygous for a whole section of your, a large proportion of your DNA, this could flag this up. Now the question is, what would you do with that data? Clinicians are going to obtain this data, and clinicians may well find out that, a child is 25% homozygous, which strongly suggests that there's been an illegal incestuous conception there. So this is the sort of data that could come up which are completely unanticipated when people enter this sort of genome sequencing trial. So the question is, which of these are reasonable to disclose? And I suppose there's no correct answer as to which of these are reasonable to disclose, but people who enter into projects such as the 100,000 Genomes Project will sign a disclaimer at the beginning that will say whether they want to be informed of findings that were unanticipated. So if you are entering into a trial for, I don't know, an ocular degeneration type disease, retinitis pigmentosa, for example, you enter your DNA, you enter your family's DNA into this. If they find a mutation in that gene or something that causes that disease, they will let you know. If they find a mutation in BRCA1 or 2, they will only let you know if you tick the box to say, I want to know about incidental findings. If they find a mutation in BRCA1 and you're genetically predisposed to cancer, but you have elected not to find out about these incidental findings, you will not be told. In terms of this bottom one here, I suppose that would be looked at from the point of view of duty of care, and decisions will be made that are way above my levels of explanation of what the cut-off is there. But if someone is in a vulnerable situation, I'm sure that data will be acted upon accordingly. Now, I've talked quite a bit about genetic diseases and what to do with data and how that would potentially be used. The cancer side of the study, and we'll talk more about cancer genetics next time, but patients who have their entire cancers sequenced, that will give us data that might predict whether patients will or will not respond to therapies. So, for example, there are mutations like the V600E mutation in the BRAF gene. Melanomas often have this mutation, and there's a drug that specifically detects, not so much detect, that specifically inhibits the mutated form. So this is where we can use genomics in a really positive way to give specific drugs that work against specific mutations. However, there are other drugs such as Herceptin and cetuximab. These are monoclonal antibodies, very expensive. They bind growth factor receptors, and there are lots of mutations downstream, so in RAS, RAF, PI3 kinase, all the things downstream of those growth factor receptors, and if they're mutated, they might mean that the drug won't work. And you can imagine a situation where a patient has their cancer sequenced and they're on course to have something like Herceptin or cetuximab, but then the genome data comes back and we have all of these mutations that predicted to possibly, maybe, not quite sure, stop this drug from working. So I suppose you could argue there might be the ethical considerations there. Should this data be used for patient management in that way of effectively withholding a very expensive drug on the basis of genetic data which is not being, let's say, wet lab tested, that we have not proven that this mutation stops this drug from working, but theoretically it might. So this might be one area where there'd be concern. They might be using this genomic data as an excuse for not giving a potentially beneficial, very expensive drug on the basis of a mutation that doesn't have experimental validation. And this is what clinicians are finding themselves in this position quite a lot now. We're finding lots of genomic mutations, somatic mutations in the cancer, and that might predict. It's been very useful for predicting what drugs will work, but when it's a lot of this data is saying that some of those drugs won't work. So there are serious ethical considerations to think about there. Okay, that's probably enough from me for one session. In the live session I can talk more about the sequencing technology if you want me to. I can talk about different ethical dilemmas that clinicians will face with handling this data. I can talk about other examples where the bioinformatics and the need for better bioinformaticians, or bioinformagicians as I call them, we can talk about situations where they are extremely useful for handling this type of data